Hi, I'm Chloe Wohlenberg, and I'm a fifth year PhD candidate in the Air Bear Lab at UC Santa Cruz. Here, I study how the cell targets and degrades problematic mRNAs, a process called quality control. Our cells all go through this process of turning DNA to RNA to proteins, which are the critical building blocks of our cell. I'm particularly interested in how the cell turns RNA into proteins, a process called translation. Translation involves these cellular machines called ribosomes, which turn messenger RNA into proteins. However, not all translation goes perfect, and sometimes ribosomes will encounter various obstacles on the mRNA, all of which can lead to improper protein production. Dysregulated protein production is a hallmark of aging and disease, and so understanding how our body manages these bad mRNAs is important to develop potential therapeutics. Thankfully, the cell has a pathway called no-go decay to respond to a particular type of obstacle I'm interested in called a ribosome stall. This stall slows ribosomes and gives time for a trailing ribosome to catch up and collide, and eventually leads to the recruitment of a factor that initiates no-go decay, resulting in the bad mRNA being degraded. Our lab is interested in this overarching question of how does no-go decay properly get triggered to degrade problematic mRNAs? If we can understand this question, we can understand how this pathway can be altered in disease. My thesis work started with a prior graduate student in the lab identifying this factor called CAPI6 to be required for no-go decay in a nematode called C. elegans. CAPI6 is a protein that transports a small molecule called a polyamine into the cell. And what's really interesting about CAPI6 is that the human version is causative of Parkinson's disease. But together, this set up the question of my thesis, which is how is CAPI6 required for no-go decay? What I found in my research is that CAPI6 transports polyamines that are important for the maturation of this important translation factor called EIF5A. EIF5A gets this special modification which helps it bind to the ribosome and help ensure proteins are made properly. Altogether, my work has led to a new model for how the cell may target and degrade mRNAs experiencing ribosomal stalling, where EIF5A and ribosome collisions act together to recruit no-go decay factors and elicit mRNA decay. My work has shown that EIF5A is required for no-go decay activation and expands our knowledge of how the cell responds to problematic translation. Furthermore, polyamines, EIF5A, and CAPI6 all have a variety of known roles in aging and neurodegeneration, suggesting that dysregulated no-go decay could be an unexpected consequence of these disease states. This opens the door to identifying new drugs to invigorate EIF5A or no-go decay as potential therapeutics. Thank you.